Hello, everyone. My name is Tuan Nguyen, and I'm a senior informatics consultant at CSols. I've been working in the laboratory informatics space for the past 20 years. I started out my career as a lab analyst and then moving into LIMS support development and currently providing consultant services for LIMS. I have extensive experience around the laboratory workflows and very good in understanding the laboratory processes in various industries, especially manufacturing, foods, clinical, and public health. My current focus right now is doing STARLIMS development and software data management information system, SDMS. Data innovation that allow the instrument integration with STARLIMS and is powered by Abbott. My hobby, I love fishing and gardening. So just a quick overview of Cso and Our Services. We specialize in North American consultancies. Our focus is solely on the laboratory informatics. Our consultants are very experienced in laboratory backgrounds. We offer services to support LIMS strategy, implementation, and validation. As a partner of Abbott Informatics, we are able to ensure our excellent services for our customer. So now I'll pass it to Carla to give us an introduction of herself and an overview of today's agenda. Carla?
Thank you, Tuan. Hi, everyone. Very happy to be with all of you during this webinar. Carla Paulsen. I've been with Starlings for almost 20 years, with the last 10 being Abbott Informatics. I have been involved in numerous LIMS implementations for public health and clinical laboratories mainly. I've been under various roles, business analyst, project manager, product analyst, and now I'm the senior product manager for the Starlings Life Sciences solution. I have my MBA and also a certificate in biomedical informatics. Also, I'm PMP certified, and my background is on computer science and software development. And then during my spare time, I'm a busy mom of a 10-year-old boy. I enjoy working out and playing tennis. So a quick overview of Starlings, Abbott Informatics overview. So you probably have heard of Starlings, that's the brand name, and originally Starlings was founded in 1986. It was acquired by Abbott in 2010, and it became Abbott Informatics Division, with Starlings being the primary solution product suite. So Starlings integrated solution is actually a combination of different Abbott Informatics products, but always having the LIMS as the centerpiece. So all of these other products or solutions are Sdms, ELN, advanced analytics, and mobile solutions. So for today, the agenda of our webinar is going to start with the business needs of this implementation that we did in partnership with CSols. I'm going to go over specifically in the process of sample reception, aliquoting, storage, and testing. Then I will explain about the clinical studies, the concept of this aliquoting plan specifications that were required for this customer. Then Tuan is going to go over the solution of the batch aliquoting that was implemented, also going over the sample storage and distribution for testing. So this will be all part of the overall workflow solution. And at the end, Tuan will also cover the instrument interface implemented as part of this implementation with BioMicroLab. So let's start with the business needs of this implementation. So just to give you a little bit of background about this customer. So this is a biopharmaceutical organization. This laboratory does testing and provides services to the various clinical trials, clinical studies that they perform, and they have to receive samples from multiple studies on a daily basis. So there is a central lab reception area that takes care of that. So first thing that we have is that these samples, they arrive with a requisition form that has the information of the clinical study, the visit, the subject, so a lot of information regarding, the sample that that has arrived to the lab and needs to be received. So these samples are received, and once they are received, they are loaded in this instrument, BioMicroLab. What BioMicroLab does is basically checking the weight, the volume of the samples, and it will provide the information of the sample volume for each of these samples. We will have this integration as part of the solution with the Starlings so that this sample volume can be captured. So imagine now we have knowledge of the samples that arrived, and now we know also the volumes of each of the samples that arrived. Now, the next thing that this team in the central lab reception area, what they typically have to do is looking at the different clinical studies, because these samples could be coming from multiple studies, and each of these studies have different protocols. There is this concept of Aliquoting Plan. So they will typically batch and group the samples for, let's say, the same clinical study and visit, so that they can take a look at this Aliquoting Plan and follow that Aliquoting Plan for that group of samples. They will manually do this aliquoting according to that plan, and once they complete that, there could be some samples remaining from the parent samples, and so if that's the case, those will need to be stored, and also for the aliquots that have been prepared, they will need to also be stored. Once they are completed with all of this storage, the next thing is going to be the transferring or distribution of these aliquots to the different testing groups. And so this is something that we also need to take care of, because we have to make sure that we capture that whole chain of custody of the samples. So the aliquots were created, the aliquots were stored, and then the aliquots eventually are picked up by, let's say, user John Doe from testing group A. So doing all of this manually, and especially when you have to handle high sample volume, can be a very daunting experience when you have, so many samples that you need to aliquot. So that is where we come, and we have to now not only understand, see what is the whole process the business needs, and see how can we address this with the Starlings implementation. So for that, first of all, I want to go over the whole concept of the Starlings clinical trials. We have a trial manager solution in our system that basically will help with this whole process, because part of the clinical study is typical that we have to take care of specifications depending on the study protocol, such as the study schedule. And the schedule is typically composed of these time points or visits, or also bleed numbers. So this is going to be, based on the protocol. For example, a study can have, let's say, 5 visits from these patients, and we will also know what kind of samples are expected to be collected and sent to the lab. So things like the specimen type, the container type, we will know that. So for instance, in visit number 1, we're expecting to get blood sample or plasma sample or serum sample, etc. So we also, need to be able to capture that as part of the schedule of the study. But also there is this concept of test panels, because many times we not only receive just the samples to, let's say, be stored, but we need to perform testing on these samples. So it could be one or it could be multiple tests. So that's something that we already have as part of our Starlings Trial Manager application. So just to give you a visual, this is our Trial Manager application, and we manage here our clinical studies. There is a lot of information around clinical studies that we manage as well, but just to highlight the key information that we need to capture in this case, things like the study ID or the time point, the visit or visit number are available in this master schedule. The tests that need to be performed are available as well here, and information like the expected sample type is also available in our master schedule in our containers test. Now, for this case, with this customer, there was this concept of this Aliquoting Plan. And so looking at that, what was missing in the Starlings out-of-the-box solution was this information. And even though we have for each of our study schedules, the information of the test name, and we know also the testing group that is going to take care of this test, a specific test, we didn't have the information of the aliquots and the priority number, which was very important to have, because part of the plan could be perhaps to do 3 aliquots, and knowing the priority will allow the user then to do in the right order, because maybe the 3rd aliquot could not be done. Maybe there was not enough sample volume. So priority number was important to capture. Also, the sample volume that we needed to allocate for each of these aliquots was another important piece. So I highlighted here in red information that we needed to capture and we didn't have in the out-of-the-box solution. And also to give you a better visual of what this Aliquoting Plan for this customer look like, I mean, this could be put into an Excel spreadsheet, which is the way a customer was managing this prior, to this implementation. It's just basically like a table where you have, based on your clinical study, for instance, study XYZ, I have a time point, bleed 01. I am expecting that for this time point, the samples that I'm going to get from the subjects of the patients, the participants of this study, will be aliquoted 4 times. My priority number 1 aliquot will be 700 microliters and will be used for test dengue 1 and dengue 2. My priority number 2 aliquot will be for flu HA1, and it will have a volume of 300 microliters. Then my priority number 3 aliquot will be flu HA2, also 300 microliters. And my last aliquot will be Zika, for Zika testing, for 100 microliters. But that is not enough. As you can see here, you can tell this is the information for time point bleed 01 for this study XYZ. Now, even within the same study, now going into time point bleed 02, let's say we have another visit that we're expecting, the second visit of the same study protocol. We may not have necessarily the same list of tests. So notice that here, instead of Zika test, I have now to do ECL test. And also, the sample volume is not something that we can manage at the level of the test, because this can vary also by a study and time point. So in the previous one, I had for my first aliquot that was used for dengue 1 and dengue 2 test 700 microliters. In this case, I would have 400 microliters to be allocated. And the same for the aliquots 2 and 3, I have a 200 microliters instead of 300 microliters. And the last aliquot would be for testing ECL, and that would be with 100 microliters. So this is very something that will give you, a visual of how, this aliquot, what it looks like, and how then the users will use this information when it comes to them aliquoting, all of these samples that they receive on a daily basis. So with that, hopefully now that gives you an idea of the aliquoting plan, but there is also another concept that we need to introduce, which is the type of the samples. In this case, this customer was managing primary and retention tubes. So what exactly is a primary tube for them? For each study and time point, basically it was expected to be at least one. There was going to be one primary tube. And the retention tubes were more a kind of like a backup tubes that were used for purposes of retesting if it was needed to do any retest, or if, for example, there was not enough sample volume on the primary one, then we could use sample, from the retentions, to do aliquoting for any other tests that were needed. Another thing that was also part of their logistics, these primary and retention tubes, even though they were collected from the subject or participants at the same time, for that one thing, study time point, they were not shipped to the lab in the same shipment at the same time. And the reason for that was to reduce risk in case that a shipment was lost or something happened to it, then chances of having another, sample from that patient, from that study participant, then, we will have at least, the chance that another sample will be available at a later time. So that's the reason why these primary and retention tubes, this could be, sent in different shipments. So we needed to do enhancements to our Starlings out-of-the-box solution for this customer. And the final solution, one of the things that we needed to do is define this aliquoting plan. So for that, just to give you again the visual of our trial manager application, we had to do these enhancements in partnership with CSols. And I'm just highlighting here where you can find, this is our master schedule where we defined all of the different time points, components that we have for this schedule. And we already had the ability to define the panels that needed, the test panels that needed to be tested for each of these time points. But what was missing was the aliquoting plan. So we added this tab, aliquoting plan. And as you can see, this looks very similar to that table, to the tables that I just showed you that the customer was managing before in just Excel spreadsheets, that gives you the information of the aliquot number, the priority number, the test, the volume that needed to be allocated for each of these aliquots. So this is going to be then the information that will be used then eventually for the execution of these aliquots. So that covers the aliquoting plan definition. So now let's say, okay, great. Now I can also define the plan in Starlings. And that's important, right? Because if we need to take care of the whole aliquoting preparation, Starlings needs to know that aliquoting plan, but now we're going into the workflow of preparation of these aliquots. So this whole aliquoting workflow, we needed to do some enhancements also to our solution. So the final solution to be delivered to this customer needed to be not only take care of supporting high volume to do this batch aliquoting, but also the user interface needed to be very intuitive and user-friendly. We needed to provide a quick way to view all of the pending list of samples that needed to be aliquoted in the right priority order, and allow the user to make edits after they completed the aliquot. Basically do a reconciliation of what's the theory, the plan versus the reality. And at the end, store any samples left over from the parent samples and the aliquots, and have all those aliquots ready in boxes or containers for the lab to pick it up. So in Starlings, we do have this workflow engine application that allows users to configure workflows that have multiple steps, that allows to process our samples for any kind of maybe preparation and assays that require multiple step preparation. So we use that solution for this, but now in the context of aliquoting. So we had these following steps. Number one, we started with a select samples step where we were allowing the user to create a batch run for samples to be aliquoted. Then in step number two, we would have those samples that were selected as part of my batch or run. We would be able to define the plan. And this is where the whole aliquoting plan would come to play, because they needed to look at that plan and select at the end the ones that they will be aliquoting. Step number three would be the preparation and confirmation of these aliquots, because this is where the user will start doing the manual aliquoting according to the plan. And if there are any deviations, then they will just reconcile, be able to edit, and confirm the final outcome of the aliquots. Step number four would be now to store the parent BIOS, which is basically the parent samples, the ones that came. If there is any sample left over, then those will be stored. And also for the aliquots, they would need to be also stored. So that's step number five, where we have these aliquots stored in some boxes or racks, containers, so that they will be ready for lab to pick it up. And that would be basically the last step. So now I'm going to pass it on to Tuan, who's going to go over the user interface that was implemented for this batch aliquoting solution for this customer.
Thank you, Carla.
Tuan?
Thank you, Carla. So as part of the development process, based on the business needs that Carla has identified, we meet as a development team collectively identify the data model and the design to support the aliquoting workflow. So for the step one, as you can see here, the end user can open the aliquoting workflow application and select the study to create a particular run for this study. On the left-hand grid, the user will have a list of samples available for them to select to create the run by moving the sample to the right-hand side grid using one of the add button option here, or remove from a particular run. There's also additional filter to allow the user to narrow down the list of samples specifically need to create for the run. Once the samples are assigned and ready to create the run, end user can click on Create Run at the upper right-hand corner button here to create a unique run ID for the group of samples have been selected to perform aliquoting. User can also click on the next step, or the workflow will bring the user automatically to the next step. We'll see the step number 2. The interface for the user for step number 2 allow the user once again select a study if they close the application and come back in, or if they click on the next step while they're on the same application, then the study and the run ID automatically populate. In this step, as you can see, the user have a data grid here that allow the user to view the Aliquoting plan based on the configuration for the Aliquoting plan defined in the trial manager that Carla has gone over. From here, user can determine if a sample meet the criteria to create the aliquot, and also allow to mark the particular sample can be performed aliquot. Some of the specific configuration, such as the amount required and the test panel for each of the aliquot supposed to be performed, will also be listed here. And the total amount currently available for the sample and after aliquot, the amount after being aliquoted. So if the user's okay with the aliquot plan here, he or she can click on Start Aliquoting to begin the aliquoting workflow. And that will bring us to the next step, step number 3, to be prepped for confirmation. So from this step, the user have the additional action where they can add more sample, they can readjust the parent quantity if any changes to the amount of the parent sample, or perform the recalculation as a whole for all the sample being selected for the run. In addition, the user can also print labels for the aliquoting sample or vial tube, or generate a pick list. The pick list can be a report, or in this case here, the pick list is a CSV file that can be imported into an instrument like Synomic for identify the barcode of the label. Once the list of samples have been confirmed and ready to complete the aliquoting process, either through manually or automatically, the next step will be allow the parent sample to be moved to the storage, while the aliquoting sample can be moved to the transfer box. So we'll go ahead and go to step 4 here. So as you can see, for the parent sample for that particular study and run, the parent sample, which is the primary sample here, can be moved to a storage. The user can select an existing box with the capacity that available to add additional tube, or the user can create a brand new box and assign the parent sample to a particular started location. For the aliquot, they'll be moved to the step 5, which allow the user to prepare the transfer box. So here we at the step 5, which is the last step of the aliquoting workflow. The user, once again, can select the study and the testings for that particular aliquot panel. Based on the list of sample, user can create a new box to store the aliquoting sample and assign a location that can be available for pickup. And finally, just a quick screenshot showing the standard STARLIMS interface, allowing the user to move sample to a particular container. So the next part of the, as part of our agenda, I'd like to go over the STARLIMS integration for our BioMicroLab instrument using Sdms. So as Carla mentioned in the beginning, the sample being received at the lab and then place or put onto the BioMicroLab instrument to scan the barcode and measure the weight for each tube. The BioMicroLab will export a files with the simpler format here as an example. The SDMS grabber can monitor a particular network share drive and pick up these files, and then process and parse the data, then look up the Starlings biorepository database and find our matching sample. If we find a matching sample for those tubes or the barcode, we will update the external barcode in the MicroLab in our biorepository, as you can see here. So this process allows the lab to automate the reception of the sample without human error. And this process can handle hundreds or thousands of samples at the same time. As part of this effort, we also create a new application. We call it file import log, because SDMS queue might be reserved or need access at the admin level to view the log or any issue with the file or doing the data validation. The application file import log here allows the end user to select a particular file type and any file that has status error based on the date range. The user can see the import for the particular file. You see the file name. And if any error or discrepancy with the data, the end user can be notified and resolve accordingly and re-upload the file if needed to correct those data. So I believe that would be conclude the technical section of the presentation. I'll hand it back to Carla to give us a summary of the benefits for our project.
Thank you, Tuan. Thanks. So I hope you all have learned about this whole workflow that was implemented for this customer. So again, the batch aliquoting workflow, this was a key process for this customer. They needed not only to be efficient because it had an impact on the turnaround time, but also because at every lab, they want to minimize out of stability samples. So that is basically when the sample already has been in either room temperature for a longer period of time that cannot be used for testing anymore. So always lab is trying to not waste any of these precious samples from these clinical trials. And also because this is the very first process. So in the sample reception, we cannot have bottlenecks that will impact, the overall turnaround time because that needs to be completed so that the samples can be then distributed to the different testing groups. And you may be thinking that why this laboratory is still doing all of this manually when there are solutions in the market for taking care of all of these aliquoting automatically. So example of that is the Hamilton. And actually this customer also uses that. But one of the things that I gotta mention is that these are instruments that are very expensive. And even though the customer has a Hamilton, that was covering only 50% of their sample volume that they needed to handle on a daily basis. So of course, as every lab, these sample volumes typically keep growing and growing. So bringing just another instrument sometimes is not just a easy or right away solution to take care of this. Plus, also as every instrument, there is always the risk for having the instrument down for multiple reasons. Sometimes it's also maintenance, and the laboratory cannot afford, just because the instrument is out of service, we cannot process, these samples, we cannot receive these samples and do the aliquoting. So as a conclusion, they always need to have a solution for doing this aliquoting manually, and they call it batch because it was such a high volume that doing it individually was not very efficient, of course. So they needed to have all of this in batches by the starting and the time point. And then another thing I just want to mention is that the Aliquoting Plan enhancements that you've seen that I show in the trial manager, all of these enhancements we are planning to also bring that feature in a future life sciences Starlings product release. So in case we have other customers with similar needs, then that could be also addressed with this Aliquoting Plan. And now to finalize, I think it's important to highlight the partnership benefits. So always keeping in mind that, with the customer success as our combined focus, Abbott Informatics and CSols provide targeted expertise to deliver the technical solution to meet the customer's needs for their Starlings implementation. CSols Starling Services team ensures the highest quality of services execution best aligned with Abbott Informatics standards and practices. So now I'm going to hand it over to Megan to do the Q&A. Megan?
Thanks. Thank you, Carla. Thanks, Tuan, for your presentation. So just a reminder, if you would like to send some questions over, you're going to use the questions panel on the GoToWebinar. And just a reminder, we will be sending out an email with a link to this webinar recording. All right, so first question here is, what STARLIMS product and version is the Aliquoting Plan workflow have been developed and tested on?
Carla, I can take this question if you want.
Mm-hmm.
For our customer, we implement the Aliquoting workflow on the version 11.1.3 clinical or life science product.
Okay. Mm-hmm. Next question. What other instruments were integrated to STARLIMS using SDMS for the Aliquoting workflow?
As part of the project, we had two other instruments. One is the genomics instrument that allow, as I mentioned in the Aliquoting workflow, user can export a work list in CSV format and import into the genomic instrument. And once the genomic instrument read the barcode of the Aliquoting tube, we check in the CSV file with the update barcode and then update our biorepository. So that is another instrument, genomic. The other instrument Carla mentioned is the automatic Aliquoting instrument called Hamilton. So that's also allow the capture of the latest volume from the parent and the volume from the aliquot in biorepository using SDMS.
Thanks, Tuan. All right, looks like that is all the questions that we have right now. If you think of any questions, you will have my email address. Feel free to reply back to me and I can get those questions over to Carla and Tuan. We thank you for your time and joining us today, and we look forward to seeing you at future webinars. Thank you, everyone.
All right, thank you. Bye.
Thank you. Bye-bye.